All right. Hi, everyone. Thanks for joining us for our February Research Reels webinar. So as always, I'll get started with a little bit of an introduction before we go into our speaker. So to start off, I'm going to introduce IMPART to you as always. So IMPART is a national research team headquartered at Dalhousie Medicine, New Brunswick. And although our headquarters are located at Dalhousie, we actually have partners and collaborators all across Canada. So this encompasses 45 academic and clinical partners across 8 universities. And we are continuing to branch forward, and we have lots of funders and other stakeholders as well. So Research Reels is a monthly seminar that we created to improve networking amongst IMPART members, enhance opportunities for future collaborations, and support knowledge translation of research findings. So you'll see today we have a wonderful speaker lined up, but if you know someone that would be interested in presenting in a Research Reels webinar, stay tuned to the post-webinar survey and you can nominate a speaker yourself and maybe see them in an upcoming seminar. So IMPART is present on all these different social media platforms. As you can see, we're continuing to grow. We have a website as well where you can learn more about some of our members. And we also upload some of these Research Reels webinars to our YouTube channel, which is Research IMPART. So you can take a look here. Some of our past seminar series have been uploaded, as well as our different conferences and meetings. So we have the Holiday In Party up here as well. So please feel free to share our YouTube channel and also subscribe. Let's get into the fun stuff. So I'd like to introduce today's speaker to you, which is Emma Holjak, a Master of Science student at the University of Guelph in the Department of Human Health and Nutritional Sciences. She's under the supervision of Dr. Jeremy Simpson. So you can also find more about the Simpson Lab on our Simpson Lab website, which is down below here. So Emma focuses on research that investigates and characterizes cardiac physiology and pathophysiology in a range of preclinical models. Before her master's degree, Emma completed a Bachelor of Science in Biomedical Science, also at the University of Guelph. And today Emma is here to present her talk titled, "Can Natural Selection in Animal Models be Used to Identify Novel Therapeutic Targets for Heart Failure?" So I will now pass over the floor to the speaker herself.
Thank you for the introduction, Arena. Okay. All right. Okay, everything looks good, Irina? You're the only one I can see. Okay, perfect. So yeah, my name is Emma Holjak, and today I'm going to be talking to you largely about the first part of a recent project myself and my lab has completed, but also about our next steps and where we're hoping to take the project in the coming years. And the title of my research talk is, "Can natural selection in the SHR colony be used to identify novel therapeutic targets for heart failure?" So to start off, I thought I would go over what heart failure is, as this is the disease in which my project focused on. So heart failure is a chronic progressive condition in which the heart muscle is unable to pump enough blood to meet the body's needs for blood and oxygen. So basically, the heart cannot keep up with its workload. Initially, when heart failure onsets, the heart tries to compensate by either enlarging the ventricle chambers, developing more muscle mass, or pumping faster. And this occurs as your heart is trying to find ways to increase its output. However, none of these compensatory actions are long-term fixes, and eventually the heart failure will worsen, and these processes will no longer work to maintain adequate blood or oxygen output. So now that we know a little bit more about heart failure, how is it caused? So there's a wide range of conditions that can damage or weaken your heart and cause heart failure, some of which include coronary artery disease, faulty heart valves, damage to your heart muscle, inflammation of the heart muscle, or myocarditis, and important for my research project, high blood pressure, also known as hypertension. If your blood pressure is high, your heart has to work much harder than it should to circulate blood throughout your body. And over time, this extra exertion can make your heart muscle too stiff or too weak to properly pump blood. So next, I thought it would be interesting to kind of highlight some of the prevalent statistics on hypertension-induced heart failure, as this is the subtype of heart disease that my project focused on. So this disease is one of the more prevalent conditions of the developed world. In Canada alone, over 6 million adults have high blood pressure, and worldwide, over 1.1 billion adults have this condition. Additionally, hypertensive heart disease alone is responsible for 25% of all causes of heart failure. Specifically, within the disease or condition of hypertension, 10% of adults have secondary hypertension. And this is high blood pressure caused by another disease, such as kidney disease. but the overwhelming majority, making up 90% of individuals with hypertension, have primary or essential hypertension. And this means that they have hypertension with no known secondary cause. So their hypertension is believed to be due to a complex interaction of an individual's genetics and their environment. And I thought this was really interesting because the model that we worked with for this project is actually one of the only animal models for essential hypertension, and that is the spontaneously hypertensive rat. So to start off with a bit of background information, the SHR, or the spontaneously hypertensive rat colony, was originally developed in 1963 by Kyoto University investigators. This colony was obtained by inbreeding a spontaneously hypertensive male Wistar rat with a female Wistar rat that had a slightly elevated blood pressure. Rats were deemed hypertensive if they had a blood pressure greater than 150 millimeters of mercury. And after 3 generations of successive breeding, 100% of the rats were hypertensive at 15 weeks old, and the SHR colony was created. So at this point, you might be wondering why the colony was created in the first place. Well, prior to the development of the SHR colony, there are very few ways to study essential hypertension. And upon establishing this colony, the investigators noted the potential of the colony to be analogous to human essential hypertension. And this was really exciting because it would, one, allow investigators to actually better characterize this condition, but also it would provide a way to test therapeutic treatments for hypertension and its syndromes. So additionally, due to the systemic effects of their chronically elevated blood pressure, the SHR is also used to study a number of other conditions, such as ADHD, diabetes, obesity, and importantly for my talk today, heart failure. So to ensure that the hypertensive phenotype is consistently retained, SHR colonies at all institutions are maintained via inbreeding, and their blood pressure is monitored when selecting animals for breeding. Despite blood pressure being closely monitored in breeding animals, the sequelae of hypertension, so this would be things like such as cardiac hypertrophy or cardiac dysfunction that produce these syndromes, are not monitored to the same degree within the colony that blood pressure is. And as a result, it is possible that over time, the pathogenesis of heart failure has been bred out or diminished in the colony. So cardiovascular fitness is an important component of sexual health, as improvements in cardiovascular health has been associated with an increased breeding rate and a greater quantity of successful pregnancies. Thus, it would be beneficial if the SHRs have adapted to better tolerate their chronic hypertension. Unlike humans, the SHRs develop hypertension at their reproductive age, so the age in which the animals are being selected for breeding, and this provides the opportunity for natural selection towards adaptive traits for hypertension-induced heart failure. Furthermore, given the debilitating effects of chronic hypertension and that the SHR colony was established over half a century ago and is maintained via inbreeding, there is a strong possibility that natural selection has caused the evolution of the SHR's original phenotype to produce a healthier animal. So therefore, the objective or purpose for the first study that we did was to determine whether the SHRs have evolved over time to the cardiac sequelae of hypertension. And then given that the cardiovascular health is impaired in the SHRs at the time of breeding, we hypothesized that over the past 60 years, the SHRs have adapted to better tolerate their chronic hypertension. In order to investigate this, we performed a systematic review of the literature followed by post hoc statistical analyses to support the notion that through natural selection, the SHR model has drifted. In order to fully characterize cardiac health within this colony, articles were acquired based on the following 3 methodologies of interest. So that was morphometry, echocardiography, and invasive hemodynamics. Articles were identified using a key term search of the PubMed database, and predetermined inclusion-exclusion criteria were created. In order to account for potential differences in methodologies such as environment, equipment, and individual investigator technique, control data was also analyzed from the Wistar Kyoto rats, and these were the rats that were used to establish the SHR line through selective breeding, and also the animal that is commonly used as a control for the SHRs in the literature. So ideally, the time frame for each methodology, which are those arrows you see spanning the timeline, would kind of perfectly line up for each category, so the morphometry, echocardiography, and hemodynamic function. However, given the differences in each technique prevalence and establishment, and the need to maintain a sufficient time period of separation, which we chose as greater than or equal to 10 years between the initial and current colony groups, these date ranges between each of the 3 investigated techniques varied slightly. However, as a result of these accommodations, we were able to collect sufficient data to perform a statistical analysis. So for each of the graphs going forward, each data point represents the mean value for said parameter obtained from a previously published study, meaning that each data point that you see on the graph actually represents anywhere from 5 to 15 rats. And yeah, so the SHR colony was originally established with the purpose of creating a genetically stable model of hypertension. However, whether the severity of the SHR's hypertension has remained stable over the past 60 years has not yet been assessed. So in order to evaluate this, we compared systolic blood pressure, or SBP for short, between the initial and current colony SHRs. In this graph, systolic blood pressure is on the y-axis and the age of the animals is on the x-axis. Additionally, the initial colony SHRs are depicted in black and the current colony SHRs are depicted in red. And just again, the initial colony SHRs are what we are calling SHRs from studies that were published in the 1970s, 80s, and 90s, and the current colony are from the 2000s onward. So as you can see from the graph, blood pressure increases from 5 to 20 weeks of age and plateaus thereafter at around 195 millimeters of mercury. Together, this data demonstrates that the severity of hypertension within the SHR colony has remained stable over time. And this was a pretty expected finding because, as I mentioned at the beginning of this presentation, blood pressure is the one thing that is really controlled for by institutions that maintain this colony. So it was expected to find that this was well intact over time. So hypertension causes compensatory cardiac hypertrophy, which aims to normalize wall stress to that of a normotensive heart. Often the magnitude of the acute hypertrophic response is driven by the severity of hypertension. So in order to investigate whether the SHR's hypertrophic response has remained stable over the past 60 years, we evaluated heart weight in the initial and current colony. Body weight was evaluated alongside heart weight to ensure that changes in heart weight were not the product of fluctuations in body weight over time. While body weights were unchanged in the current colony SHRs, as shown on the graph on the left, current colony SHRs showed significantly increased heart weights. Additionally, no changes were found in body or heart weights in the Wistar control rats. Thus, changes in the current colony SHRs were unlikely due to differences in environmental factors such as changes in animal housing standards over time or feed. So overall, these results indicate that although the severity of hypertension has remained consistent within the SHRs over time, current colony SHRs undergo greater hypertrophic remodeling. In order to further investigate changes in cardiac structure and function, echocardiographic data was assessed. Current colony SHRs showed a decrease in posterior wall thickness, which is indicative of concentric hypertrophy of the left ventricle. Despite a decrease in mean stroke volume, which typically accompanies concentric hypertrophy, mean cardiac output in the current colony SHRs was maintained. This response is likely due to a compensatory increase in heart rate, given that cardiac output is the product of stroke volume times heart rate. To ensure these hypertrophic changes in the SHRs were not due to external factors, echocardiographic parameters were also assessed in the initial and current Wistar control rats. And both cardiac structure and function were consistent between all control animals. So collectively, these data further demonstrate that chronic hypertension in the current colony SHRs resulted in a more heightened hypertrophic response. Previous findings have established that increased cardiac hypertrophy results in greater left ventricle wall stiffness, which leads to elevated diastolic filling pressure and diastolic dysfunction. While echocardiographic data or echocardiography is often used as a non-invasive measure of cardiac function, invasive hemodynamic assessment is the gold standard. So to understand the functional ramifications of cardiac remodeling in the current colony SHRs, several invasive hemodynamic parameters were assessed. Overall, there was no significant differences in any of the indices of cardiac contraction, which you can see in the DP, DT max graphs on the left, or there was also no differences observed in the indices of cardiac relaxation, which you can see in the graphs on the right. Again, the red are the SHR graphs, and then the two graphs below are for the Wistar control animals. So since no differences were observed in any of these indices, we were able to conclude that the increased hypertrophic response did not cause changes in cardiac function in terms of contraction and relaxation. So what does this all mean? Well, here we show that while the severity of hypertension remains stable in the SHR colony, the cardiac sequelae of hypertension has changed over time towards greater concentric hypertrophy. This work establishes that concentric hypertrophy is beneficial in a hypertension setting when developed through natural selection. Together, these data firmly establish a phenotypic shift in the SHR model for the first time since its initial establishment in 1963, and provides important insights into the adaptive hypertrophic response for hypertension-induced heart failure. So what are the broader implications of these findings? Well, as I mentioned at the beginning of my presentation, heart failure is a global health concern that affects millions of people each year. This study provides the first step in being able to understand and utilize the adaptive hypertrophic response to hypertension-induced heart failure that was naturally selected for in the SHR colony. And currently, we are actually in the process of continuing on with this project in its second step. So the next steps after we published the paper that overviewed the data I just took you from, we published that probably in the fall is when it actually came out. And what we've been doing since is locating researchers who potentially have SHR tissues from what we called the initial colony. So that again is SHR hearts from the 1970s, 80s, or 90s. And our goal was to find researchers who worked with this animal and had tissues either saved in the form of processed for histological analysis, so most likely saved in paraffin blocks, or stored in a -80 freezer. Because once we had our initial colony samples, then it's more easy for us to order in current colony SHRs, as those would just be SHRs that are currently getting bred and sold through companies like Charles River. And then we can fix those hearts ourselves and process them for analysis. The end goal is once we have tissue samples for both groups, we can run a comparative tissue microarray analysis between the initial and current colony. And what we're going to be looking for on that microarray is which molecular pathways are involved in the hypertrophic adaptation we found in the SHR colony. And then by better understanding these molecular pathways, or which molecular pathways natural selection has selected for, we can potentially harness this mechanism and design a therapeutic target or some sort of therapeutic that could be used in the management of hypertensive heart disease in humans, which is really exciting. And we've actually been ongoing with this project for the past few months. So we've already been gathering what we're considering is our tissue bank of SHR tissues from previous researchers who worked at it. However, we are still in the process of collecting more tissues because we are basically just happy to get as much tissues as we can, because the end goal is if we have enough, we could study the SHR's molecular pathways over time and maybe do a decade by decade analysis. So my Twitter handle will be and my email at the end of this presentation. And if you yourself know anyone who worked with SHRs in the 1990s, or potentially you know of someone else from a different institution, feel free to email us, as we are still actively collecting tissues if people have them. So with that being said, I just want to give a big thank you to my advisor, Dr. Jeremy Simpson, as well as everyone in my lab for their overwhelming support and their intellectual contributions throughout the course of my studies. And with that being said, thank you all so much for listening. I'm happy to answer any questions. And yeah, my email and my Twitter are just below my name here if you know anyone who has any SHR tissues.
Thanks so much, Emma. So I'm now going to stop recording.